Okay, so welcome once again. In today's class, we are going to look at the key points. We'll start our series on histology key points. Specifically, in this lecture, we are going to consider the epithelial tissues. We are going to consider the epithelial tissue, and if you remember, how many types of tissues do we have? How many types of tissues do we have? Okay, so we have four, right? And those four tissues um, include, you have epithelial tissue, you have muscle tissue, you have connective tissue. Which other tissue have I not mentioned? I've mentioned three. I have mentioned three types of tissues. I've mentioned epithelial, connective, so epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Okay, so we have four types of tissues. Okay, muscle, nerve, connective, and epithelial tissue. So we'll begin today by revising epithelial tissue. Okay, so some of the general considerations as far as epithelial tissue is concerned is that when we're talking about the general consideration, we have classification. Okay, so we have classification of epithelial tissue, and basically we have lining and covering epithelia. Okay, we have lining and basically covering epithelia. Okay, so now this lining and covering epithelia basically just forms the boundary between the external environment and the body tissues. Okay, so this covering and lining epithelia basically just covers body surfaces. For example, the epidermis of the skin. Okay, and it apart from covering body surfaces, it also lines the lumens of internal organs. Okay, apart from covering body surfaces, this lining and covering epithelia also lines body cavities. For example, peritoneal cavity, and it also covers the exterior of the organs which project into these very cavities. Are we together? So, um, apart from that, you need to understand that the cell shape and the number of layers of this covering epithelia basically correlates with the function of the epithelium. Okay, so the function of this particular lining and covering epithelia is directly related to the function of that particular epithelium. Then the second classification of epithelial tissue is what we call glandular or secretory epithelium. Okay, so this glandular or secretory epithelia basically just develops from a lining or a covering epithelium by invagination or proliferation. Okay, so what happens is that it can either form when the invagination has occurred or when the proliferation has occurred, we can either have exocrine gland or endocrine gland. Okay, I repeat on glandular epithelia. What I'm saying is that the glandular epithelia basically just develops from a lining or a covering epithelium via invagination into the underlying connective tissue or proliferation. Okay, so when the invagination has occurred or when the proliferation has occurred, basically we are going to have either exocrine gland or endocrine gland. Are we together on that one? All right. Okay, so let's consider some general features of all epithelial tissues. So these are just general features of all epithelial tissue, either whether endocrine, I mean whether glandular or lining epithelia. These are just general features of all epithelial tissues. Okay. Number one, epithelial tissue is highly cellular. Okay. Highly cellular. Um, where what we mean, we're talking about sparse intercellular space. Okay. Sparse intercellular space. Then another general feature is that we have numerous intercellular junctions. Okay, we have numerous intercellular junctions. Okay, so we have numerous intercellular junctions. So these numerous intercellular junctions are there basically for for anchorage. Okay, for anchorage. Then another thing that you need to take note of is when we talk of epithelial tissue is avascular. Okay, epithelial tissue is avascular. Are we aware of what avascular means? Okay, so when we are talking of avascular basically just refers to an area where there is, number one, there are no blood vessels. Okay, number two, since there are no blood vessels, there is insufficient blood supply. Okay, number three, um, there is lack of vascularization, which is just the formation of blood vessels. Okay, so when we narrow it down to anatomical context, we're just talking about a tissue or an organ with limited or no blood flow. Okay, we are good. All right, so another general feature is that epithelial tissue basically has high regenerative capacity. Okay, has high regenerative capacity, especially in epithelial membranes, in order um, to replace continual sloughing of cells from free surface. Okay, so what do we mean by this statement that epithelial tissue has high regenerative capacity? What we mean is that high regenerative capacity basically has to do with the ability of cells or tissues or organs to rapidly recover from damage or injury. Okay, or basically just to efficiently repair or replace damaged cells, and thereby quickly returning to normal function and structure. Okay. So another general feature is that most of these epithelial tissues, they rest on what we call basement membrane. Okay, they rest on what we call what basement membrane. Okay, so when you are talking about this basement membrane, you need to understand that the basement membrane basically, how can we put it? The basement membrane has the basal lamina. Okay, the basement membrane has a basal lamina which has lamina lucida. So the basal lamina comprises of three things. You have the lamina lucida, you have the lamina densa, and you have the lamina reticularis. Okay, and then this basal lamina has some additional components, okay, which include pelican or pelican. Okay, so the basement membrane, apart from having just basal lamina, also comprises reticular lamina. We are good. All right. Now let's begin by let's talk about the lining and covering epithelial tissue. Lining and covering epithelial tissue. So when you're talking about the lining and covering epithelial tissue, um, one of the things that we can consider has to do with classification. Okay, so we can classify these covering epithelial tissue by the number of layers. Okay, by the number of layers. So as far as number of layers are concerned, we have what we call simple epithelium. Okay, so this simple epithelium just basically has is one cell thick. Okay, we have just the one cell layer. Okay, so simple epithelium just has one layer. Okay, so you'll find that all the cells all the cells will rest on the basement membrane. Okay, all the cells will rest on the basement membrane. Okay, then we also have stratified epithelium. So stratified epithelium from the word strata. Okay, or from the word or we can say more than two layers. Okay, so this is a kind of covering epithelia which has more than one cell layer. Okay, and since it has more than one cell layer, what will happen is that only the layer which is the deepest in that in those layers will be in contact with the basement membrane. Okay, and you'll find that those which will be on the most superficial cells are the ones which are going to face the free surface. Okay, so that is classification by the number of layers. We have simple and stratified. Then we also have classification according to the shape of the surface cells. So we have squamous. Okay, we have squamous. So these squamous, basically, the cells are much wider. Okay, much wider than tall, and they resemble a fried egg. And then you'll find that their nucleus, the nucleus of the squamous cell, is highly flattened. Then apart from that, we also have cuboidal. So cuboidal cells, this this cell is basically is in equal height. You'll find that if I can just draw something, you'll find that this cell it is something like a cube. Okay, something like a cube where you find that the height and the width are in equal, they are equal, okay? Then the nucleus of the cuboidal cells is spherical, okay? The nucleus is spherical, okay? Then we also have columnar. So on the columnar cells, just from the word columnar, from the word column, you'll find that these cells, they are much taller um, than the width, okay? So the height is greater than the width, and their nucleus is oval shaped, okay? And this nucleus is generally located towards the base of this particular columnar cell. Are we good? Are we okay with that? Let me know if you are able to, if you are following. All right. So these are the types, okay? So when it comes to the types of lining and covering epithelia, we have simple squamous, okay? We have simple, okay? I go back a bit. What exactly should I reiterate on? Okay, on the shape. So I was uh, I was saying that when it comes to classification of these lining and covering epithelial tissues, one of the classification is done by the shape, by considering the shape of the surface cells. Okay, so we have the squamous. So the squamous, basically, the squamous cells, these cells are much wider. Okay, they are much wider, whereby uh, we can compare them or relate them to a fried egg. Okay, and then their nucleus is flattened. Then we have cuboidal cells. So the cuboidal cells, from the word cube, okay, from the word cube, meaning these cells are of equal height and width. Remember your cube, okay? And then the nucleus of the cuboidal cell is spherical, okay? Then we also have columnar cells. So these columnar cells, from the word column, so you'll find that if I were to draw just something roughly, this will be the columnar cell, okay? This will be the columnar cell. So you'll find that the nucleus of the columnar cell will be oval, okay? The nucleus will be oval. So the nucleus of the columnar cell will be oval, okay? And apart from being oval, it will be located at the base of the cell, okay? So it will be located somewhere here at the base of the cell, not on top, but somewhere here. Okay, the nucleus. Are we good on that one? All right. So now let's talk about the types of lining and covering epithelium. Types of lining and covering epi epithelium. Okay. So we have simple epithelial tissues. Okay. We have simple epithelial tissues. So when it comes to simple epithelial tissues, um, these which are on top, okay, these ones, these are simple, they belong to the group of simple epithelial tissues, okay, simple epithelial tissues. So when I'm talking about simple epithelial tissues, these are tissues these are epithelial tissues which are not stratified. Okay, they just contain a single layer. So under such category, we have what we call simple squamous. Okay, so simple squamous basically allows for rapid diffusion across the epithelium. Okay, so simple squamous allows for rapid diffusion across the epithelium. Then you'll find that this simple squamous normally forms the lining of blood vessels, alveoli of the lungs, and internal body cavities, because it allows for diffusion. Remember, in the alveoli of the lungs, that is where you have a gaseous exchange taking place, and it takes place via diffusion. So hence we need simple squamous epithelium there. Then we have simple columnar. Okay, we have simple columnar. So as for simple columnar is here. Okay, we have simple columnar, as you can see. So the simple columnar basically, it lines and also it is involved in absorption. Okay, it is involved in absorption. So this simple columnar basically forms the lining of the intestines and the gallbladder. Then we also have pseudo stratified. Okay, we also have pseudo stratified. So when it comes to pseudo stratified, the cells, as you can see, um, as you can see right here, you'll find that the cells are of various heights. You can see that, right? The cells are of various heights. Okay, is this visible? I'm sure you're able to see that the cells are not in unison. Okay, so you'll find that all cells rest on the basement membrane. Okay, but only the tallest cells will reach the what? Will reach the free surface. Okay, so the variation in height of the cells and the location of the nuclei give basically the appearance of a stratified epithelium, of which usually it is frequently ciliated. So pseudostratified epithelium basically normally provides protection, and it forms the lining of the respiratory tract and much of the male reproductive system. Okay, we are good on that. All right. Now let's talk about because okay, we have this other category on the bottom here. I haven't talked about this. Okay. So now, um, we have the other category which belongs to the stratified epithelial tissues. Okay. Stratified epithelial tissues. So under stratified epithelial tissues, we have um, stratified squamous. Okay. So stratified squamous basically protects from physical abrasion, and it also prevents uh, uh, dissection. Okay, not dissection, no, dissection. Okay, and under squamous, stratified squamous, we have two types. We have stratified squamous non-keratinized. So this non-keratinized, we can say stratified squamous moisturized, but that won't sound anatomical. So it's better we use stratified squamous non-keratinized. Okay. So this non-keratinized stratified squamous normally lines the wet cavities. Okay. Lines the wet cavities, um, including the mouth, esophagus, the rectum, and the anal canal. Okay. Then we have stratified squamous keratinized from the word keratin. Okay. So this one, in other words, we're talking about a dry stratified squamous epithelium. Okay. So this normally is found in the epidermis of the skin. Okay. The epidermis of the skin. So you'll find that as far as the stratified squamous epithelium is concerned, the surface cells are non-living. Okay. So apart from that, apart from the stratified squamous, we also have stratified cuboidal or columnar. So these ones, they normally line the larger ducts of exocrine glands. Okay. Then we have this unique epithelium known as transitional epithelium. Okay. So the transitional epithelium has a protective function. Okay. So the protective function is that this transitional epithelium is basically constructed to expand as far as distension of the hollow organs it lines is concerned. Okay. So to distend, I believe, is how can we put it? Is it to increase in size? Okay, in a layman's language, to increase in size, and then at the end again come back to normal. Okay, I believe you understand what I'm talking about. All right, so this transitional epithelium is basically unique to the urinary system. Okay, as it lines the urinary bladder and the ureter. We are good. All right, we proceed. Now let's talk about surface specialization. Let's talk about surface specialization. Okay. So the surface of epithelial tissue basically gets specialized. Okay. Surface of epithelial tissue gets specialized. Okay. So what happens is that um, this surface specialization includes, we have microvilli. Okay. So microvilli basically, what are these microvilli? Microvilli are just finger-like extensions. Okay, these are just finger-like extensions um, from the free surface of the cell. Okay, and these microvilli, basically, they are present in large numbers on each cell, and they are collectively are called brush or striated border. So one of the key components is that these microvilli, they contain actin microfilaments. These microvilli contain actin microfilaments. And these microvilli are relatively non-motile. Okay? Are relatively non-motile. And the microvilli are very much prominent on the cells which are lining the digestive tract and the proximal tubules in the kidney. Then, apart from microvilli, we also have stereocilia. Stereocilia, these are just large non-motile microvilli, not basically cilia, no. Okay? So, stereocilia also contains actin microfilaments. Stereocilia also contains actin microfilaments. Okay, so stereocilia basically is present on the cells which are lining the epididymis, okay, and the ductus deferens, specifically in the male reproductive tract. Okay, then from there we have cilia. Okay, so as you can see, microvilli and stereocilia, they both contain actin microfilaments. Now let's talk about cilia. So cilia, by description, we can say that these are just multiple hair-like extensions from free surface of the cell. Okay, so these cilia basically is highly motile. Okay, cilia is highly motile compared to stereocilia. Cilia is highly motile, okay, as it beats in a wave-like motion. Okay, so now the question is, what are the functions of these cilia? So cilia basically function to propel material along the surface of the epithelium. For example, in the respiratory tract and in the oviduct of the female reproductive system. So in the oviduct, it helps to propel the ovum. Okay. Then when it comes to the core of the cilium, the core of the cilium is basically called axoneme. Okay, axoneme, in which we have 9 pairs of microtubules. Okay, here we had actin microfilaments, here we have microtubules. Okay, so cilia, the standard cilia should have 9 pairs of microtubules. Okay, where we have this arrangement 9 plus 2 arrangement. Okay, then when the cilia has this arrangement of 9 plus 2, it will be considered to be motile. But when this cilia has it has a 9 plus 0, what will happen is this cilia won't be motile. That is the standard. So if on the 2 here you have a 0, then the cilia will be defective. Are we okay? All right.